The M phase of the cell cycle, while short, is the most noticeable part. The M phase is the fourth part of the cell cycle, and the other three phases are collectively known as the interphase. The M phase is also known as mitosis. It is the process of separating duplicated chromosomes and creating two cells. In mammal cells, the process of mitosis only takes about an hour, which is rather short compared to the other three phases. In that hour, the cells go through 6 stages. The stages in order are ProPhase, Prometaphase, Metaphase, Anaphase, telophase, and cytokinesis. The M phase is important in the cell cycle, but it requires the cell to go through the other stages before it can start. Just before mitosis starts, the cell will have already duplicated all the DNA, so all that's left is to divide. Looking inside the cell during prophase will show a few things. The DNA has condensed in the nucleus, and then microtubules from the centrosome start to form the mitotic spindle. Because a lot of these changes can be hard to see, the first change that one may notice is in prometaphase. In prometaphase, the nuclear envelope bursts, and the microtubules from the mitotic spindle will start to connect to the chromosome's kinetochores. The kinetochores are proteins on the centromere, the place where the sister chromatids connect. In the next stage, metaphase, the mitotic spindle forms 2 identical halves in the cell. In the middle, all the chromosomes are lined up with 1 sister chromatid for each side. At either end, microtubules coming from the centrosomes either connect to a chromosome or span the entire length of the cell. Those chromosomes are split in anaphase. The mitotic spindle pulls the chromosomes apart, separating the sister chromatids. The cell-spanning microtubules also push against each other, slightly elongating the cell. The M phase ends with telophase and cytokinesis. The 2 halves of the cell form daughter nuclei from the remnants of the parent cell's nuclear envelope. The mitotic spindle will further elongate the cell, forming a cleavage furrow in the middle. And finally, the 2 daughter cells will split apart going into interphase and beginning the cycle once more.